Thank you. Thank you for sticking around. It's a privilege to tell you a little bit about what we've been up to at Immunebridge. And this is the clicker. Which one do I use to advance? Is it the green one? Perfect, thank you. So we're a company that builds allogeneic natural killer cells as our first product, but underlying our company is a core technology. And it's an expansion technology that is proprietary to us. We have developed it in-house. It's a small molecule that allows us to culture HSCs, immune stem cells, from cord blood. And we believe that it introduces a novel source for immunotherapies, that's the obvious source, and that it obviates the need for iPSCs. And we believe that not only does this enable us to manufacture cells at scale, but also it gives us the choice of whose cells to manufacture, which cells to manufacture, and we believe that this matters. And so we are collaborating with cord blood banks and using our technology also as in a discovery context to unearth the more potent cancer-fighting cells within among immune systems. And as our first product, we're developing an allogeneic NK for a hematological indication, and then advancing our pipeline towards solid tumors. We're pursuing development in a very partnership-minded approach. So we have a few strategic partnerships already aligned. Some of them we will be announcing over time. But also, since we believe that what we're bringing is a capacity for making better immune cells, then we're very happy to partner with people who have engineering approaches and are looking for the right cell chassis to integrate them into, who have better disease models, organoids, or complementary therapies. And this will, and we're closing around a financing that is enabling us to bring our first product to clinic and build out this data set demonstrating that we have a better approach. This audience perhaps doesn't need an introduction to the kind of transition that we're going through and the potential for allogeneic off-the-shelf therapies. But I will reveal at least something about what are the core questions that we believe are sort of underlying this transition that we're embracing. We believe that in the context of autologous, the answer of whose cells is obvious, but in allogeneic, it opens up a broader question of whose cells. And if you're stocking a shelf of a future cellular pharmacy, you have to have an answer for how you're going to manufacture. And this isn't just a bioreactor question. Cells don't like dividing ad infinitum. You need to understand the biology of expansion. And NK cells, of course, they're natural killers. They have a panoply of features, including the capacity for tissue residence, the capacity for recognizing heterogeneous malignancies. But the question still is, which of those features and how can you amplify them? And how can you combine both the understanding of the biology and then also with whatever other engineering you layer on top of that? So it opens up new questions for engineering. And so for these three questions, these are our answers. So whose cells? We believe that genotypes matter, that NK cells are meaningfully different from human to human. And we have an approach, which I'll show you, to systematically screen and then translate those cells rapidly. So we screen cord blood units and identify the optimal donors. For manufacturing, we believe that the obvious source for building immune cell therapies is from immune stem cells. And for that, this is the proprietary technology that we can take a cord unit and expand it at scale. And then we take the produced expanded HSCs and differentiate them to potent NKs. And in terms of the features of the NKs, we screen them across a variety of attributes in order to understand them on a per indication basis. And then on top of that, we have the capacity to then take the better cells and then engineer them further with a variety of approaches. And so this part, the screening and the expansion, is uniquely enabled by our technology. But with regards to further enhancements, this is the part that we're open to partnerships. So end to end, what does our process look like? It has these 5 steps at the input of our process as a cord blood unit. And we're collaborating currently with the New York Blood Center. And we have an approach for how we pre-select them, and I'll mention a little bit about that. We are currently pursuing both an engineering and a non-engineering approach. But one of the things, the points that we're trying to convey with this expansion is that, and it obviating the need for iPSCs, is that we have a similar capacity to add engineering at the low cell volume stage. So immediately after isolation from a cord blood unit, then expansion, which we do for, it's a choice, it's a dial that we can tune for how long we choose to do the expansion in order to stock a master cell bank. But for reference, we get about 10,000-fold expansion in 6 weeks of HSCs. From that master cell bank, we would take a seed train and differentiate them down to NK cells or other immune cells, which are then bagged, cryopreserved, and this can achieve a large scale. So, have we built out data around our expansion? Have we understood its reproducibility? Yes, we've done this now for many donors, and we've been transitioning now from lab scale to larger bioreactors and sort of packaging our CMC strategy. And so that has been actually even pushing these expansion limits even further. And so just to give a sense, what is 10,000-fold? So starting from a standard cord blood unit, isolating the CD34s there, that's actually sufficient for a very large amount of NK doses, particularly because these expanded HSCs, in the process of differentiating to NKs, further undergo expansion. And so we believe that this is a better way than using iPSCs for manufacturing cells, practically in a way that can meet market demand. And once we expand these CD34s, it's one thing to show the numbers, but have we profiled them functionally? And so by definition, if they're immune stem cells, they have the capacity to differentiate into a variety of immune cell types. So this actually represents quite a body of work of following differentiation protocols where they exist, and walking the cells down across the entire immune tree. So making neutrophils that are multilobular macrophages that gobble up beads, NKs and T cells that show the right phenotypic markers. And for our first product, we're developing an NK cell. So they're natural killers. So this is on the left-hand panel is our data where we've challenged our cells with a variety of hematological malignancies, and we've benchmarked them against published data from a variety of companies. And those are the cells that are currently have been rolling off of our pipe, and we're enhancing them further. So this is the non-engineered one. I mentioned that we can engineer early and expand, and I mentioned that we can differentiate into a variety of cell types. And so while our focus is primarily on building NK cells, we decided to build out this data as a proof of concept, where we've expanded the bank of HSCs, demonstrated that the modification that we introduced was persistent, and we differentiated both into the NK and then also into the myeloid lineage, and saw antigen-specific killing in the NK stage and nibbling in the macrophage monocyte context. And so just to summarize, what is it that we have? We have the capacity, we're starting from a novel source. It gives us the optionality to select donors. Then it allows us to choose what developmental stage to walk them down into, because these are cells that are readily differentiable into natural killer cells. And we can choose to make maybe earlier progenitor of NK cells that can have beneficial homing or persistence features. These are some of the hypotheses that we're testing. And also, because the same source is multipotent, it allows us to combine cells, and as we've demonstrated for the RNKs and CAR macrophage. But what I haven't told you yet is what is it that makes donors different, and what do we believe in how we're intending to plumb that. So when I think of an NK cell, I think of as a logic-gated cell, so to speak. It has a variety, dozens of activating and inhibitory receptors that are rather sophisticated at smelling out different kinds of malignancies. And these arrays of receptors are meaningfully different from human to human. I like to think that HLA is the most diverse locus within human diversity, and the second most diverse is KIR. And so whereas HLA is expressed across all of our bodies, nature is telling us that there's something meaningful about these innate cells, which do a lot of the early cancer fighting among us. And so some of the differences in these alleles are known. There's some data sets that have been proven out. All of this is based on clinical data, but not all of them have been screened and tested systematically. And so this is what we're doing. We are selecting donors that represent the different flavors of NKs that exist among humanity, and then we are challenging a variety of cancers with them. And this is not our data, but it's sort of part of the inspiration to convey one point is that when you look at CAR NKs, even a CAR negative NK kills, right? And you don't necessarily see that with a T cell. So if you have plus minus, you see killing and you see no killing. But when you look at, this is a paper that is a HER2 CAR NK, and you can see that when you adjust the axes, that you have one donor that has CAR negative has more potency for killing than someone with a CAR positive. And so this is just making a point, is that there's a contribution to the efficacy that comes from the cell itself, and then there's, on top of that, is engineering. And so this is that choosing donors is important. Another sort of longitudinal study from the Cancer Genome Atlas that we're inspired by tracks people across a variety of indications, and then had sequenced them and bin them into the kinds of NK genotypes that they were born with. So you can see that 8 to 10 years out, you have a huge survival advantage for the, in this Kaplan-Meier curve, for the favorable NK genotype. And if this was a drug, this would be a blockbuster drug. But this is just the roll of the dice of the NK cells that these people were born with. So the obvious question is, can we take advantage of those features and then help those that are in the less favorable population advance? So how do we do that? And so this is, so this is sort of the premise, and I'll show you exactly how we're doing this. That better donors allow more potency with less editing, and we have a genotypic hypothesis that is inspired by past clinical data, but we need to empirically prove this out. And so this is kind of the core of what we're doing. This is what a cord unit looks like, and that thing is called a noodle within the industry. And it has very few immune stem cells in it, but we have an expansion technology. And so what's great about a cord is that it's actually a licensed product, right? And when we talk about off the shelf, there is a shelf that has lots of these meaningfully different immune systems in waiting. And we can sample them from these cords and manufacture enough in an end-to-end run to run all of the preclinical studies that we need, including animal studies. And that actually preserves the actual unit untampered for translation. And what this means is that something that has been the bane of the field, like donor-to-donor variability, actually for us is a strength. Because yeah, donors are different. Humans are different. There are some that are more adept and more suited for certain kinds of challenges and tasks. And in this case, it's for cancer killing. So this is our process. We screen, which we pre-select based on our genotypic prior knowledge, expand, differentiate, and run the natural killer cells that come off through a panel of assays to identify the favorable features. This both identifies our, enhances our knowledge, connecting genotype, phenotype, and function, but it also empirically then identifies the product. And so these are some of the technologies that we're working with to establish process control between lower scale, higher scale, and also the scaling out for the arrayed screening. This is, don't need to look into this too much, but this is conveying a little bit of how we think, that we're generating a meaningful matrix of distinct therapeutic candidates, and then assaying them across a variety of meaningful disease models. So this is using the old sort of chemical compound library and updating it for the cell therapy world. And in this process, we're generating an abundance of data and trying to systematically understand what are the different features of cells. And so we're doing this both with non-engineered cells in order to identify their capacity, and then also layering on engineering in a purpose-driven basis. And so when we think about our development strategy, that one of our differentiating aspects is the capacity to identify the optimal genotypes. And so where is there the data, where is there still an unmet need and there's data around that a genotype meaningfully matters? And so that's why for a first indication, we're working on an unmodified NKs for AML. And we're also simultaneously working on a variety of engineering strategies to further potentiate ourselves with trying to line our sights up on solid tumors. And we also got a DoD grant. So for the current kind of strategy, development roadmap strategy is we have this platform, but we want to use it to be on path towards the clinic. The point is we have a manufacturing approach that is actually practical. And so we want to keep that in mind. And so currently we're focused on building out the pre-IND data set, develop our manufacturing plan. We've been building out our team, these core capacities of CMC, assay development, and R&D. But at the same time, as I mentioned to you, we're building out this discovery platform and data set demonstrating that we can make superior cells. And where we hope to be in about a year and a half is on the cusp of doing our first GMP run for the clinical materials and progressing towards an IND. And we hope to be able to share with you in the future this data set that will come out of this work that will demonstrate the features and the capacities that will make other people want to collaborate with us in co-development partnerships and in other ways to make great therapies. And so overall, kind of thank you. This is just to highlight that at this meeting we have a few members of our team. You're meeting me, Nina, our head of NK therapeutics research is in the audience. Ru is the head of manufacturing. Nina's work was around NK cells and tissue trafficking. And so we're very happy to have her on board with this very important work. And we also have some industry veterans on our board, including Kim Warren and Greg Bonfiglio, and then Alicia Jackson was previously also a DARPA program manager. And yeah, I'll leave off with kind of our mission, and it's important to us because we believe that not only are we making therapies, but we're making them scalable, and we're keeping in mind the COGS for manufacturing them, because we want to make these curative therapies accessible to all, and we're being humble in front of nature and learning from the solutions that are banked within human diversity.